Thanks everyone for coming. If you can, even for a moment, imagine yourself standing here and in this position, you'd know how extraordinary it feels and how fortunate I feel to be recognized in this way. Thanks to the Academy for their efforts and Hans and Ove for their introductions. What I want to do today is tell you a little bit about this uh, humble fish and some of the work that is ongoing in my lab to try to address questions on the origins of species. And by species, I mean the evolution of reproductive isolation, the evolved characteristics of species that prevent them from interbreeding, either because they don't recognize one another as mates at all, or alternatively, if hybrids were ever produced, then they would be less fertile or viable. The question I want to use to frame this talk is one that was raised by Dobzhansky soon after the modern synthesis. And he was asking what kinds of genetic differences underlie the origin of species. And in particular, is it the case that the very same genes that underlie the evolution of phenotypic differences between populations, beak size, body shape, are they the same genes that are responsible for reproductive isolation? Or is it the case that there are special genes beyond those responsible for species differences that underlie reproductive isolation and that become established only under special conditions? So we do know that for some cases, especially pre-mating reproductive isolation, that the same genes must underlie reproductive isolation as are responsible for phenotypic differences, because the phenotypic differences involve traits like the time of reproduction. And so when populations evolve different breeding times, then automatically as a side effect, they would interbreed less and less often. But it remains a question, particularly for postzygotic isolation, by which I mean the reduced fitness of hybrids, reduced fertility, reduced survivorship of hybrids. And the sort of central genetic model that sort of underlies this question is one also proposed by Dobzhansky and independently by a couple of other people, Bateson and Muller. And the idea is fairly simple. Imagine two populations beginning at the start of this time sequence are genetically similar at two loci, A and B, that over time an advantageous substitution occurs through the origin and spread of the capital A mutation, which replaces small a, and then subsequently a capital B mutation replaces small b. And in the ancestral population across the same time period, no change occurs. The idea that Dobzhansky proposed was that when big B and little a are combined for the very first time in a genome of a population, that something happens, some malfunction occurs. Signaling protein can't talk to receptor, and the result is reduced fertility and or reduced viability of hybrids. So he proposed that there are genes that have these effects, and that they would likely be the ones to underlie postzygotic isolation. And this model has even been put into practice in the contemporary process of producing new species in the laboratory. So this is a paper that was published by a group at the University of Minnesota, where they deliberately followed this Dobzhansky model to create new hybrid species of flies in the lab. And the process was, again, involving two stages. First, the manufacture of a mutation A on a line of a lab line of flies, and then secondly, the manufacture of the mutation B in the population that already contained big A. And what B does is it drives overexpression of some absolutely important gene, but if you carry big A, you're resistant. And the consequence then of mating the big A, big B line with the little a, little b line is that all the embryos die. I find this philosophically interesting, but also raises questions about natural speciation and what we can learn from artificial speciation about the way new species originate. But mostly I want to talk about natural speciation today. So again, Dobzhansky's question, what kinds of genetic differences are involved in the origin of species? And he proposed that the way to answer this question was to study the genetic basis of the isolating mechanisms encountered in nature. And this has been done now in a number of different taxa, including flies and mimulus plants. And the basic idea is to try to identify the genetic changes that are responsible for the reduced viability or fertility of hybrids. And what's also true of these studies is that, as Dobzhansky proposed, they usually involve interactions between pairs of genes. So from the previous model, a big B and a little a. But through this work, very little has been discovered regarding the connection between the evolution of ordinary phenotypic differences between populations and the genetic changes that cause this reduced hybrid fitness. In fact, in most examples where a story can be proposed to explain the spread of these mutations, they have been attributed mostly to processes other than those that cause species to become different from one another, and instead to various forms of intergenomic conflict, such as meiotic drive, and then also reciprocal loss of duplicated genes, involving a process whereby a gene is duplicated, it's transposed to another chromosome, and then in two different populations, each of them is silenced. So one of them is silenced in one population, the other one silenced in the other population. And the result, when you cross these two derived forms, is that some of the hybrids are actually missing the gene entirely. So those are processes very different from the ones that we think of as giving rise to differences in, say, beak size between populations. So I feel that we still lack a framework to connect the evolution of ordinary differences between species and the evolution of these characteristics that cause hybrids to have reduced fitness. And I'm paraphrasing Dobzhansky by saying perhaps the problem can be approached by studying divergence in ordinary phenotypic traits and then asking what is their contribution to reproductive isolation. And so that's the ongoing projects in my lab to try to address the question from this other perspective. So in outline of my talk, I've given you sort of a picture of the question, and now what I want to do is tell you something about this marvelous group of animals that has given us so much insight into some of these questions, and a bit about their origin and what we've learned about the genes that underlie reproductive isolation. It's the three-spine stickleback. So here's a map of southwestern British Columbia. The city of Vancouver is shown close to the center. And what's marvelous about this group is the diversity of forms that they exhibit in bodies of water in this part of the world. And additionally, all of these freshwater populations, which occupy lakes and streams in the region, are ultimately descended from the marine three-spine stickleback, this ancestral form which is still out there. And so it gives us an opportunity to not only examine the questions about adaptation to freshwater environments, but also to compare them with the ancestral state, because we still have it. These populations occur in lakes that are only 10 to 12,000 years old, and they occur nowhere else, and so it's clear that these forms have evolved very rapidly. Some of these populations represent some of the youngest species on Earth in anything, and this is valuable because by studying the process sort of as it's happening, we hope to be able to make discoveries. And another advantage is that they're so young that they have evolved very few intrinsic genetic incompatibilities, we can make hybrids and study hybrids. Most small coastal lakes, British Columbia, have just one species of three-spine stickleback, like this one in Cranby Lake. And to understand the origins of all of these populations, we start, 10,000 or so years ago, where my office is today was covered by ice about 1.5 kilometers thick. This ice melted rapidly. The weight of the ice had been so great that it pressed coastal lands into the sea. And when the ice melted, which it did relatively rapidly over about 1,000 years, the land slowly rebounded, lakes were formed, and they were colonized. And Derek Tan in our Biodiversity Museum made a little video which sort of illustrates this. Here's the ice melting very quickly, and then a side view of the process. The ice is melting and retreating. The stickleback are swimming in the coastal waters of the sea, but the weight of the land being removed causes this isostatic rebound, and populations of stickleback form, and they begin to adapt to these novel freshwater environments. What originally drew my attention to this group is the fact that some lakes were apparently colonized twice and possessed two almost completely reproductively isolated forms called the limnetic and the benthic. They don't have formal scientific names yet. There are challenges doing this for many of the reasons that I will describe. They're so young, it's hard to decide what Latin binomials to actually give them. With apologies to Carl Linnaeus. As I said, they're sympatric. They are almost completely reproductively isolated, but not completely. This triangle plot shows individuals genotyped in one of the lakes that has two species, and the purest individuals are deleted from this graph, and instead I just show those individuals whose ancestry is sort of more than 5% benthic and less than 95% benthic. And heterozygosity helps to identify the different hybrid classes, and so these individuals here of maximum heterozygosity are actually first-generation hybrids, and we have found that about 1.5 to 2% of the individuals in the lake are in fact first-generation hybrids, and here on the flanks of this triangle are back crosses in both directions. So hybridization still occurs, but the hybrids have reduced fitness. And as a consequence, these forms persist indefinitely, or have for 10,000 years. So again, a map of the region. A total of 5 pairs of species has been discovered, 2 of them in different drainages on this large island, Texade Island. And then Little Quarry Lake has one, Hadley Lake has one, Enos Lake has one as well. Of the 5 pairs of species that have been discovered, 2 are already extinct. And in both cases as the result of invasions to the lake. One of them deliberate. A couple of brothers introduced a catfish to the watershed containing Hadley Lake, and there are no stickleback in this lake anymore. The weird irony of that story is his name is Darwin, and he's a descendant. Here's we think that the different populations have largely independent origins. Here is a phylogenetic tree based on chromosome 15, the most boring chromosome in the stickleback genome. So very few signatures of selection on this region. And basically, the phylogeny of the populations are not so far removed from simply a star, which is what you would expect if each of the populations is colonized independently from the sea. So we have the different benthic forms in different lakes, the different limnetic forms in the different lakes. We also have single species populations, and we have the ancestral marine form. I'm shown here. The only real signal that one detects in this plot is a small branch that is shared by the benthic and limnetic form that inhabit the same lake. And we attribute this to a history of gene flow between the 2 species. So gene flow has not completely ceased even now. As I showed you, some hybrids are still formed. But it suggests that certain regions of the genome have completely independent histories, and chromosome 15 would be one of them. Some of the ecological differences between the forms, we call them benthic and limnetic because of the different ecological characteristics that the limnetic species is out in the open water feeding on evasive zooplankton, and the benthic feeds on macroinvertebrates in the littoral zone, the edges, vegetated regions of the lake, and also the deeper sediments. And we can detect those ecological differences using stable isotopes of nitrogen and carbon, shown in this plot. So isotope ratio of carbon is shown here along the x-axis, and that's an indication of whether ultimately the carbon in their diet and in their bodies that make them up derive from the littoral zone in the lake or from the open water. So the limnetics have a low carbon ratio, and the benthics have a high carbon ratio for that reason. And trophic position is indicated by isotope ratios of nitrogen, and that's because the limnetic species feeds on some of its diet are predatory copepods, and so it actually occupies a slightly higher trophic level. And what I want you to get from this plot, which I'm going to show again because I want you to commit it to memory, is that in terms of the stable isotope space, limnetics are to the left and up, and benthics are down and to the right. The various adaptations that they possess to their different environments involve muscle and bone, and also body shape. Benthics are great at suction. They open their jaws relatively slowly. They generate more suction. They have a bigger mouth. They have this muscle on the back that opens the jaw, and they're good at feeding on prey that are in the sediments or attached to vegetation. In contrast, limnetics have a jaw that is adapted for very rapid jaw opening and a far reach, which it uses to consume evasive zooplankton like copepods. And we've done all kinds of experiments in the lakes. We can, one of the advantages of working on a fish which is basically this big is that we can do transplants. We can confine each to the environment of the other and say how much better or worse does it do. And we can show here that in terms of growth, and we've shown that this is based on feeding rates, that each has a twofold advantage in the environment it prefers. Okay, so so that's a picture of the phenotypic differences between these young species, and now what is the relationship between that and the evolution of these incompatibilities that affect the fitness of hybrids. To address that, we've been carrying out studies in our experimental ponds. So here's a drone's eye view of a portion of the facility. We have 20 ponds that are each of them 25 meters in length, 15 meters wide, and 6 meters deep at one end. It's a fantastic resource because it allows us to study characteristics of behavior and morphology and other phenotypes of these populations in an environment that's fairly natural, quite different from what can be done in the laboratory. In this one experiment that I'm describing, just to give you a picture of the scale, there's me in my lab standing there. Matt Arnegaard and Gina Conte, a couple of people in my lab, and Matt later in Katie Peikel's lab, did this great experiment where they crossed limnetics and benthics, raised F1 hybrids in the lab, threw them as adults into the pond. They reproduced themselves. We don't feed them or anything. These are intended to be natural environments. And they produced an F2 cohort that then we then tracked. And we used this to map the use of the environments by their diet, and we used stable isotopes of nitrogen and carbon to do this. And you've already seen this slide, but I'm doing it to reinforce the memory in you that the limnetic is sort of top left and benthic is bottom right. And when we collected F2 hybrids from the population and measured stable isotopes on them, what did we see? Segregation variants. So we generated a range of phenotypes that extended all the way from individuals with a very limnetic diet and those with a much more benthic diet, and then all kinds of variants in between. And Matt came up with this sort of niche score, which goes from upper left to lower right and represents an axis of sort of more zooplankton, limnetic-like, to more benthic, and more benthic-like in the lower right. And then we mapped that characteristic genetically. And it mapped to over half the chromosomes in the stickleback genome, and each of those regions that contributed only a small amount. And their effects were largely, not entirely, additive, so that it was possible to predict the diet of an individual hybrid basically by counting up the number of alleles at those in those genomic regions that have been inherited either from one parent or the other. Then Matt sort of overlaid a performance surface based on the sizes of the fish at by the time they reached several months of age in the fall when these were collected. So red means very good growth, and blue means very poor growth. And the first thing I draw your attention to is this sort of saddle shape relationship between phenotype and growth. Up until this experiment, my sort of main way of thinking about why hybrids between these species might do poorly is that they were intermediate in phenotype, and therefore they would be neither best at feeding on the environment of the benthic species in the benthic environment, and it would not be very good at feeding in the environment of the limnetic parent either, and so they would suffer an advantage by being phenotypically intermediate. But when we looked at their results, we realized this is not entirely true. And that's because there's this whole class of hybrids down in this corner that in general are doing very poorly. And when we looked at those individuals, we discovered that their weird isotope signature, neither upper left or lower right, was associated with a very weird diet. They were feeding on springtails, collembola, jumping onto the water surface. And then when we measured these individuals, we realized that these were misfits of a kind that we hadn't really anticipated but should have. And that is that they represented a class of individuals that for some aspects of the jaw looked just like the limnetic species, which is adapted for rapid jaw opening and far reach. But other aspects of the jaw looked just like the benthic species, which is built for slow jaw opening and generating large suction. And our interpretation of the result was then, by being so mismatched, these hybrids could do neither of these things particularly well. They invented a novel niche in the environments of the ponds, but this did not greatly help them because on average they had the poorest growth of all. And so we began to realize that hybrids might be less fit, not just because they're intermediate, but because they're mismatched. They have weird combinations of traits inherited from the parental forms that also compromise. So some of the implications of this is that, to summarize a point I just made, which is that some recombinant hybrids have mismatched traits. And we now sort of interpret these mismatched traits as phenotypic incompatibilities, the sort of the ecological equivalent of the incompatibilities, the genetic incompatibilities that Dobzhansky described, or in his model that began this talk. These are heritable traits, and therefore there must be genetic incompatibilities underlying the mismatches that we detect in hybrids. But at the same time, I showed that these traits themselves are polygenic. They're influenced by a number of genes. And we've not been able so far to detect incompatibilities between pairs of genes underlying these mismatches. So work's still in progress, but we think that on average, most of these interactions between genes might be weak for the reasons I've stated. An intermediate phenotype in hybrids also contributes to reduced growth performance. We became very interested in this mismatch, and we're pursuing this further. And one of the projects that Ken Thompson and this wonderful undergrad in the lab of Neet Sheena did is they just sort of asked the question, is it the case that as populations become more and more differentiated, that the hybrid offspring become more and more mismatched? And the answer is yes, indeed. So here's a picture of how what mismatch looks like. Here's imagine two traits, a Gilraker number along this axis and armor along that axis. Here's the ancestral marine form. Here's a derived freshwater form. Let's draw a line to connect the two points. Any individual along this line would be considered intermediate in phenotype, but what we consider to be mismatched individual is an individual whose measurement is offset from this line. So it has some strange combination, and that would be sort of extreme in this individual here that resembles one parent for one of the traits and the other parent for the other trait, just as we saw for characteristics of jaw in our pond experiment. And what Avneet and Ken found is that as they compared populations that were progressively more and more different from the ancestral marine form, that the average amount of mismatch in the hybrids between them, both first generation hybrids and second generation hybrids, increases. So this leads to the conclusion that as populations diverge, hybrids get worse and worse through this process. So as I said, these mismatches are likely to lead to incompatibilities that so far we've been unable to detect directly by looking at pairs of genes that underlie these phenotypic differences. But we think that their cumulative effects might nevertheless be detectable, and that's a course that we're pursuing. The other feature of these incompatibilities that distinguish them from the kinds of incompatibilities that Dobzhansky, the genetic incompatibilities that Dobzhansky described, is that they should be environment dependent. So they were detected in this experiment because we threw these fish into ponds. They had to feed themselves. Whereas if we raise these hybrids in the lab, they just do fine. And so and to sort of address this possibility that there might be cumulative effects of interactions between the genes that underlie these traits, we realized that there is a prediction that emerges from this sort of cumulative effects idea, and that is that if we looked at the hybrids more closely, what we should find is that the individuals that are that do best in these hybrid populations should, on average, across the genome, be more heterozygous. And I'll try to explain with a picture why. So here's our sort of model for how populations adapt in novel environments. Here's our ancestral marine population. Again, two traits, trait 1 and 2. And here's the derived population in freshwater. And when a marine population colonizes the freshwater environment, it accumulates a series of genetic changes that gradually bring it closer and closer to this optimum in the new environment. And in a situation that whereby the effects of the underlying genotypes are additive, and in a model where mutations themselves affect multiple traits, we have this expectation of greater heterozygosity. So Ken Thompson, my student, did sort of a quick simulation of this process, and the results are shown in the plot here. So a derived population, an ancestral population, an adaptive process to accumulate a series of mutations followed by crossing. And each of these dots now represents the phenotype of two individuals, of an individual for these two traits. And Ken has colored them by the heterozygosity. And what you can see in the plot is that generally the darker dots, the more heterozygous individuals, are closer to the line. And the paler dots, on average, tend to be further away from the line. So the least heterozygous individuals tend to be the most mismatched. And that's shown again on this plot on the right, where the horizontal axis measures the excess ancestry heterozygosity across the genome in this simulation, and the amount of trait mismatch that's observed, which is distance from the line. It's hard to give you a better intuition for why that occurs than to remind you that in this process, it's assumed that mutations are pleiotropic. They affect multiple traits. And so in that case, acquiring a series of mutations that bring them closer and closer to the new optimum in the new environment, each mutation is not necessarily advantageous for all traits. It's beneficial for some traits, disadvantageous for other traits, but a net benefit. And that the process of adaptation involves the process of accumulating these pretty good mutations, the ones that are advantageous, but not necessarily for all traits. But to reach this peak, later mutations must somehow compensate for some of the sideways directions of the earlier mutations. And then when you cross the ancestor to the descendant, you create a hybrid pool where suddenly these compensations are broken apart and recombined, and you produce weird phenotypes. And those weird phenotypes are particularly apparent in those individuals that are, say, homozygous for one of the parents at one locus and homozygous for the other parent at the other locus. And that's why you produce this pattern whereby larger or more heterozygous individuals are on average closer to the line. So Ken recruited data from all of the experiments that we and our collaborators have done in the experimental ponds. And he also gathered data on lab populations that we had kept over the same time. So these are all different experiments that have been done over a period of time in by people in my group and collaborators. Some of them are limnetic benthic crosses. Some of them are marine by freshwater crosses. They're all raised in ponds. And what Ken showed is that in every experiment we've ever run, that when we retrieve the surviving individuals from the ponds, there's an excess of heterozygosity. And we don't see this in laboratory raised populations at the same time. It's not only true in every pond experiment that we've ever done, it's true of every replicate of every pond experiment we've ever done. So it's a striking pattern consistent with this idea that there might be cumulative antagonistic effects between genes inherited from this parent, and then secondarily it would be environment dependent because the heterozygosity, excess heterozygosity, does not happen in the pond, in the laboratory environment. So there are other expectations that we're now investigating as well. Another prediction from this is that in hybrid populations where one of the parent species has the majority of the genome is one of the parent species and a minority of the genome is the other parent species, there should be persistent selection against whichever alleles are inherited from the minority genome parent. So the reason for that is that in a hybrid population where one parent species is in the majority, an individual that is, say, homozygous for the minor parent allele at a locus, that individual, that locus, is working in an individual where the majority of the rest of the genome is from the majority of the majority parent, and so such alleles would experience the greatest sort of number of incompatibilities if they are present. And so we've started an experiment where we've created reciprocal back crosses and introduced them to ponds. And we will try to investigate over the next few years whether we see this major parent advantage in these experimental hybrid populations. So as I mentioned, this is ongoing work. No results for you yet, sorry. So that's a description of the ongoing projects to try to investigate really the phenotypic basis of hybrid incompatibilities with getting an assist from the genetic experiments. Now I want to talk a little bit about some other things that we've learned about the origins of these species from further genetics work. So this is a graph from a paper by Roberts Kingman et al. He was a student in David Kingsley's lab. And what this graph shows is the differences between marine and freshwater populations. And the differences between benthic and limnetic populations are similar in that. So here's chromosome 1, chromosome 2, and every dot in this image measures the degree of divergence between the the marine and the freshwater populations that are included in this comparison. It's hundreds of populations included in this comparison, both the marine and freshwater populations. And what Robert Kingman did is that those regions that show particularly high divergence are colored in red here. He called them ecopeaks. And I guess the first thing that impressed me looking at plots like these is, wow, there are quite a few areas of the genome that are differentiated between these marine and freshwater populations. Secondly, you have to realize this isn't the comparison between one marine and one freshwater population. It's a comparison between all of them. And so the peaks that you see here are those that consistently differentiate marine from freshwater populations. At the same time, I've also told you that each of these populations, or the majority of them, have colonized independently from the sea via the same ancestor. And I'm showing you that the same genomic regions are diverged over and over and over again. How can this be? The answer is that all of these genetic variants were in the marine population when these lakes were colonized in the first place. And there's now experimental evidence from Alaska which demonstrates this, which I'm not going to show. What I want to mention is that when Robert Kingman attempted to estimate the age of the genomic regions that differentiate consistently marine from freshwater populations, he discovered that on average they're 2 million years old. What? 2 million years is not just older than the lakes where these species occur. It's orders of magnitude older than the lake. There is no 2 million year old stickleback population out there. How is it possible for mutations so old to persist, to stick around this system, and then be used over and over and over again, to be present in the marine colonists of the freshwater lakes at the end of the last ice age? So obviously they brought the mutations with us, and we're trying to understand just exactly where these mutations come from. There's no lake in existence that is the source. There are no lakes that are 2 million years old that contain stickleback. And so we've come up with a sort of a model which we're attempting to investigate. And it starts with the same section that I showed you earlier, and now I'm going to describe the rest of this movie, which sort of represents our sort of a view of how we think maybe these mutations have kicked around the system for so long, so much longer than any of the extant populations which currently possess them. So again, the ice recedes, the weight removed from the coastal lands causes the coastal areas to rebound, and these lakes are colonized by stickleback, and these stickleback adapt to those environments. Here's a marine stickleback indicated in white, and many of these marine populations actually move into the bottoms of freshwater streams, and occasionally they interbreed with freshwater adapted populations that are indicated with the sort of greenish color, and creating a hybrid. And some of those hybrids may join the marine population, and each stripe represents a different gene. So that individual is sort of half fresh, half marine, but it interbreeds with another marine individual and creates another offspring, or a series of offspring that have, over time, fewer and fewer and fewer of these freshwater mutations. We have no idea how many generations occur in the sea before the next step, but try to imagine that as a result of this process, a marine population is created whereby individuals in this population possess 0, 1, 2, 3, some individuals, many of these alleles which are not advantageous in the sea, but are advantageous in fresh water. The ice recedes, the new lake is colonized, and this population brings these freshwater variants in to the new lake environment, and reproduction takes place over many generations. And in this new environment, those individuals that possess the greatest number of the freshwater alleles on average survive better and produce more offspring. And so over time, these freshwater alleles increase in frequency, and eventually, over thousands of years, recreate a freshwater population that is almost the same as one of the populations further south that may have donated the alleles to the marine population in the first place. So this is our model, and we're in the process of testing this idea. So the origin of modern stickleback species. Some post-zygotic isolation, reduced fitness of hybrids, appears to have evolved as a consequence of divergence in ordinary phenotypic traits. So I think this, at least in part, answers a question that Dobrzański proposed long ago. And this post-zygotic isolation includes not just selection against hybrids because they are intermediate in phenotype, but also because they are mismatched. I didn't talk much about phenotypic divergence and pre-mating isolation between these different populations, but that too is now known, that pre-mating isolation between them also has a phenotypic basis. The genetic basis of post-zygotic isolation involves selection on intermediate and mismatched genotypes, as I said, and many of these genetic differences are old and predate any known stickleback population. They're older than the species. So we think that progress is being made in this direction, but that there's still a lot of work to be done, particularly to connect the ordinary process of natural selection on populations, adapting populations to the environments that they inhabit, as Darwin described, and connecting that to the genetic changes that underlie the reproductive isolation that represents the sort of central characteristic of new species. And also, just to restate something I already said, the genetics underlying the youngest species on Earth might often be the result of selection on older variation. So this discovery that has been made in stickleback has been seen in other systems too, particularly other fish groups, but even in the Galapagos finches, where a mutation that arose 1 million years ago that influences beak size is now distinguishing beak sizes of species that formed only 200,000 years ago. This covered, it all looks easy. This covered a lot of years of work from too many people to list here, but here are some of the most central characters in the experiments that I described to you today, and you'll recognize this person in the audience. So thanks very much for your attention, and I'll be happy to take some questions.